Hi everyone, welcome back to the channel. This is Dr. Asi, and today's topic is DNA technology. So let's get started. So this is chapter 10 in the Orange Book, which is called DNA technology. So last topic was gene to protein, which we learn how actually genes actually converted to the protein eventually. But today is DNA technology, which is basically also called biotechnology. So if you hear the term biotechnology, so that's pretty interchangeable word with DNA technology. Okay, so let's get started. So we have in chapter 10, we have student learning outcomes, like techniques in DNA technology. So a student will learn about various DNA technology techniques, like specifically 5 major techniques we will talk about, including some genetic testing methods, and also briefly about analysis and interpretation of genetic data in this chapter. So, like every other chapter, this chapter has also vocabulary, like glossary of words that actually you can study. And when you're actually learning about this and doing some deep reading from our Orange Book, right? So chapter 10, so from the Orange Book, which is chapter 10, DNA technology. This is also called manipulation of DNA technology. Some maybe new words for you, like splicing, we learned about last chapter, which is cutting the DNA of a gene to add new base pairs in this case. Vectors, maybe transport vehicle as a vector carrying a desired gene in a host into the host cell. Plasmid, a small circular DNA found in bacteria. Restriction enzymes, special DNA cutting enzymes found in bacteria, like DNase, eukaryotic DNA digesting enzymes, DNA ligase, like that sealing and gluing things as an enzyme. DNA cloning, making more copies of a DNA amplification through use of a host bacteria cell. Restriction fragments, pieces of DNA, DNA in various lengths. Gel electrophoresis, which is the DNA, putting the DNA into a gel, moving through this gel pores, and in response to electric current. And DNA probe, which is a single-stranded DNA fragment, possess a particular sequence and a complementary to the fragment being sought. So these are some vocabulary. So let's get started. Okay, so why this chapter is important? Well, biotechnology is a very hot topic, and is actually there's a lot of new things coming out in biotechnology. Some of them we hear every day. DNA profiling, analysis of DNA samples, determine whether they come from same individual, right? DNA technology, can help find the guilt or innocence, used to confirm, like one example idea of a Boston Strangler 50 years the after fact. It's used in crop plants in biology. You hear about GMO plants. I mean, you go grocery shopping, you see GMO tomatoes or GMO corn. So they actually use a DNA technology. Human genes compared to some other genes, how distinct they are, and some other significant advances. Okay, um, so we have a number of techniques. So we have 5 DNA techniques that we will cover. One is genetic engineering here in this is chapter 10, DNA technology. Genetic engineering is one of them. Again, biotechnology is a generic term for whole DNA technology or manipulation of DNA technology. Um, so why this is done? To make maybe useful products. That's the main goal. Um, so it can date back to the many years back, using yeast to make bread or beer or some other um, selectively breeding livestocks. But biotechnology today is more like these specific techniques, like we will cover 5 of them, using this DNA technology, modern lab techniques. Okay, so GMOs is a good example. GMO is organism have acquired one more gene by artificial means. It's called GMO, genetically modified organism. If the new gene come from another organism or species, that becomes a recombinant organism, and now this is called transgenic organism. Okay, so like you see in this picture, scientists genetically engineer bacteria, mass produce a variety of useful chemicals like cancer drugs, pesticides, glowing fish like you see here with a gene from a jellyfish added to this fish and it's glowing. So there are other recombinant DNA techniques. Bacteria is commonly used, and bacteria has this bacterial plasmids, which is their like circular DNA. And so gene might be cloned there and transferred to another organism, like in plants. You can see here the bacteria inserted this specific plasmid with a gene of interest, and then once it's actually there, it's transferred to plant, in this case, like corn in this case. Or we will see insulin, for example, for diabetes people. Okay, um, so one technique is actually cutting and pasting DNA using restriction enzymes. So the restriction enzymes are they can cut the DNA and then paste them and seal them together in another combination. So DNA sequence recognized by particular restriction enzyme is called restriction site. So here is one example just showing you this how a recombinant DNA, which is actually has a piece which did not exist before, is actually cut and paste. Gene editing. This is a very new technique. This is also called CRISPR-Cas9 system. You can see here. This is maybe one of the newest, maybe only last 10 years, and already got Nobel Prize awarded for this technique. It's a very cool technique and really, really cool actually. That it's actually is a technique going and live actually editing the gene in its location. So that makes it a very, very unique. So um, it's the Cas9 enzyme. It cuts both strands of the target gene DNA and repair the enzymes randomly insert nucleotides and then knock out disabled gene. This has a lot of promise actually because of it could actually maybe used to fix genetic disorders by just actually cutting out the mutation and actually fixing it. So it's a really interesting technique called gene editing. So some medical applications of these DNA techniques, like one best example maybe will be this human insulin. So human insulin produced by a GMO bacteria right now, and it's called this brand here, Humulin. So and this actually can make it much faster and easier production of this um, insulin. And some other proteins or vaccines also now produced by these DNA techniques as well. So GMOs, back to the GMOs. GMOs basically might have a gene actually from a different organism that actually inserted in. So in the US, all corn, soybean, cotton crops might be GMO mostly. And because of that, they might be more insect resistant or higher yield or higher oil levels or some other things. So also like in rice, for example, a good example is golden rice too, which carries a gene that actually increases its vitamin A levels. That way, it's actually because vitamin A deficiency can cause blindness in children, and this actually by eating maybe golden rice too, it can actually decrease this happening. Cassava now is produced actually with higher iron and beta carotene levels, or vitamin A, which makes vitamin A. There is another technique called gene therapy, which is a normal gene inserted to a human to treat a disease by producing a specific protein. This is a clinical trial level technique and very costly, more than $2 million, maybe a dose. It's called human gene therapy to treat specific diseases. Again, this is in clinical trials. So blindness, example of blindness linked to a defective genes responsible for producing light detecting pigments in eye. A viral injection carry a normal gene, improve vision, sometimes enough to allow normal function. Just one example. Okay, so fixing blindness, hemophilia, maybe cancer. So human gene therapy technique is might be used effective for this. DNA profiling. So um, DNA profiling is analyzing DNA samples and comparing them with each other and see if they come from same individual. So this can help actually solve crimes. Maybe in a crime scene there is a DNA sample, and it says if you run it in a jail, this is how it looked like DNA profiling, and you have two suspects, and you can actually try to find which one has the most common DNA sequence compared to DNA found. So it's like a DNA match, and that can help um, eh, find out if these two DNA come from same individual. PCR, of course, another cool technique is just called polymerase chain reaction. So a PCR, you probably all heard about PCR during the pandemic, which basically you all give samples of that like to find out if if the COVID positive or COVID negative situations. So they put them in PCR and make million copies of this DNA, and then basically use this to identify DNA profiling or other stuff related to this. Okay. Bioinformatics is another technique, which is basically DNA sequencing is an example, applying computational methods to store and analyze biological data, which is called like DNA sequencing. So DNA sequencing getting cheaper and faster every day. So now there is a technique called next gen sequencing, which is basically um, can actually cut in the fragments of 300 nucleotides and then thousands or hundreds of thousands of the fragments sequence simultaneously, and then sequence more than 2 billion nucleotides a day finally. And then there's a third gen sequencing, a very long DNA molecule sequence its own. So these are some new bioinformatic techniques. Genomics is not the newest technique, but it's also an area in DNA technology, which is actually studying the complete sets of the gene, not just one single gene, and then try to see specific patterns. Like you see in this picture here, we have actually sets of genes of Arabidopsis plants and rice plants and corn plants and chimpanzees and lab rats and human and a lot of others. There is a technique called genome mapping, which is basically, it's like the name suggests, to map all the chromosomes and all the known genes on every chromosome in specific locations. And then the genome mapping techniques can be also like whole genome shotgun method, just one way to do that. And then finding DNA sequencing and deposit them in gene banks that's open to the whole public. So there was Human Genome Project, which is in 2000s and completed, and now we know there are 21,000 genes actually in human genome. And also 2000 disease associated genes identified, including Behçet disease, life-threatening illness, swelling of blood vessels. There are some ethical questions also raised by DNA technology. There might be dangers, some dangers might be, of course, this needs to be studied. Like disease associated genes could they be abused? In response, U.S. Congress passed Genetic Information Non-Discrimination Act in 2008. Title 1 prohibits insurance companies requiring gene info during health insurance application. So any genetic info. And Title 2 provides similar protection for employment. So there is one chromosome, it's very small chromosome called Y chromosome in male, which is basically direct father's line ancestry. So human Y chromosome passes essentially intact from father to son. So that makes it really interesting to find out information like 8% of the males in Central Asia may be descended from Genghis Khan because of Y chromosome reveals that information. Another example is 10% of Irishmen descendants of this Niall of 9 hostages, a warlord of 1400. You can see the Y chromosome here, the small chromosome with 76 genes. So there is a summary of this, basically to just to recap, this is chapter 10, DNA technology or biotechnology. We talked about 5 major DNA techniques, including gene editing and GMO plants and human gene therapy and DNA profiling and forensics. So do some more deep reading from chapter 10 and complete the quiz, and see you next video. Thanks for watching. As always, please feel free to comment, subscribe, and like. Happy studying.